Alright, so as we discussed before, the cell is always going to try to, thermodynamically, things always try to reach equilibrium. The cell sort of disrupts that equilibrium so that it can use the energy to carry out certain cellular processes. Everyone follow that? Yes? Some people are nodding. Yes. So, who can tell me some of the important ions that function in the cell? Could you list some? We already talked about a few. Sodium, potassium, sorry? And hydrogen in plant and animal cells, yeah? Yeah, hydrogen, yeah. Something else? Chlorine, that's an important one. Someone said sodium, yeah, you said sodium. There's one more really important one, calcium. Okay, that's the one. So these are the molecules that you usually focus on. They're going to stay in equilibrium across the membrane. I don't know if I need to write it down. What I want you guys to look at is a particular experiment that could be done. So what is the concentration of sodium outside of the cell? Let's say that this is the outside of the cell, extracellular. What do we call this? Medium, matrix, or whatever it is. Does anyone know what the concentration of sodium is generally outside of the cell, in other words, in your blood fluid or something? Pardon? It's going to be 150 millimoles. Okay, this is the concentration of sodium. And inside of the cell? 47. Where do you guys maybe I should stop asking number questions. 15 millimolar. So, I dare not ask. What's interesting about these numbers? They mentioned something very interesting in the textbook. We talked a lot about evolution, things like that. I guess you guys didn't read, but what they were saying is that this concentration is very similar to the concentration of sodium in the ocean. So that's pretty cool. Why is that cool? Anyone like evolution? Shows that we evolved from the ocean. Things probably crawled out of the ocean, and then as we were evolving, the body already had mechanisms in place to use this molecule, and it said, okay, we're going to bathe all of our cells in something very similar to the ocean. Is that cool? Does anyone think that's cool? Who thinks that's cool? Right, half the class. The other class, you're haters. You hate Mother Nature. So this is sodium. The concentration of sodium. We'll also look, let's say, at the concentration of potassium, of course, K+. All right, so let's say that this is a semi-permeable membrane. So these molecules are charged, right? Potassium has a positive charge, sodium has a positive charge. This is going to be the source of energy for a lot of processes that take place inside of the cell, right? You can couple a lot of reactions to the movement of the molecules down their electrochemical gradient. So in your book, you'll see a diagram where they sort of attach what's basically a voltage meter across the cell, and they can measure the potential difference. So what is the resting potential of your cell membrane? I heard a few things. I think it's about -70. Negative meaning that it's negative inside or outside? Inside. So inside of the cell is generally negative, which means most of these molecules are moving up. And the body will try and maintain this difference, okay, of -70. One of the things you put in your study guide is -70 millivolts a lot?
For a cell. Maybe for a cell. So
it's not only for a cell, but actually the diameter of the biological membrane is 3, I'm already forgetting, it's 3 or 9 nanometers. It's 3 or 9 nanometers. It's nothing, right? It's super small. But if you talk about 70 millivolts per cell, the equivalent to that is about 200,000 volts per centimeter, right? Can you imagine 200,000 volts passing through that? It's actually quite incredible. And yeah, I thought that was interesting. So, So the importance of this experiment, I think you want you guys to know the Nernst equation. And the point of the Nernst equation is how can you measure the potential difference across the membrane if you know the concentration of ions on both sides of the movements. So what would happen if I put a sodium channel in this membrane? Where is the sodium going to move? I even put the arrow. I should have put the arrow. Some of you guys are still looking at me like I'm in outer space. Obviously, sodium is going to move from the area of high concentration to low concentration. So you're going to get a certain voltage potential that you would be able to theoretically calculate if you knew the concentration on both sides. So that equation is going to be called the Nernst equation. It's Na plus K equals. Look in your book for the details of this. I'm not going to go into it. 591 log 10. And it didn't ask him if you guys had to memorize this equation. But you probably will. Does he let you guys use sheets in your exam? Here's a good way to study, and Dr. Zergis taught me this. Write down as many notes as you can on one sheet of paper. Try and fit everything on one sheet of paper as if you were allowed to bring it onto the exam. And you'll find that once you get to the exam, you remember everything. That's helped me a lot. Okay. So this will calculate the electrochemical potential across the cell if you know the two concentrations, okay? This is the equation you may or may not need to know. So the electrochemical potential equals 0.0591 log 10 over the concentration of sodium on the right side of the membrane over the concentration of sodium on the left side of the membrane. Okay? Or you could look at the outside to inside, for example. If we're talking about a cell, it would be the outside to the inside. And again, these are the kind of energetic reactions that would cause the you can use it for antiporters, for symporters, because naturally, if you use an ATP pump now, so if we look at this as the inside of the cell and the outside of the cell, we could use an ATP pump to move these back here, right? So it's kind of like a fool's errand. They're just moving these things back, but they just want to go there, and we can generate energy using this. All right, let me try and turn this back on. Sorry, guys. So are there any questions so far?